Hello, I am Khanna, an assistant professor at the Department of Ophthalmology at UMass Medical School in Worcester. My work relates to understanding how defects in our light-sensing cells, called photoreceptors, result in severe blindness disorder. And one of the major blindness disorders here is Leber's congenital amaurosis, or LCA. Now, as you all know, LCA is a severe form of blindness that occurs early in life, which is also called childhood blindness, and it results from the defects in our genes. My particular interest is in understanding the defect due to the gene called CEP290. Now, forget the technicalities, but defects in CEP290 result in a death sentence for photoreceptors. I am particularly interested in designing treatment strategies for the type of LCA that is caused due to CEP290. Now, there are two ways to treat these disorders. One is to replace the dying cells with new and fresh cells, and you may have heard of this technique, which is called stem cell therapy. This is a new and upcoming technique and holds a lot of potential. The second one that we work on is to understand how the defect is caused, and we can fix the defect. In order to fix a defective gene, we could replace it with the correct version of the gene. This is a very attractive approach and has immense potential, as is known from the recent human clinical trials for another form of LCA that is caused by defects in a gene called RPE65. However, there is a limitation to the size of the gene that can be delivered through this approach. CEP290 exceeds the size that can be packaged and delivered into the cells. So in such a case, we have resorted to fixing the defect by using alternative approaches. What we are trying to do, and this work is all funded by partly by donations from such events there where you are participating. In this case, first of all, we try to identify how CEP290 works in photoreceptors, and then identify the defect where it has stopped working, and then we try to fix that defect. The second approach that we are trying to do is to identify smaller versions of CEP290 that can be packaged and delivered into the dying photoreceptors. We are now testing the potential of the smaller parts of CEP290 to serve as alternative approach to delivering the full CEP290 gene for gene therapy. In the end, I would like to say that our light-sensing cells are extraordinarily complex structures, but nature has devised ways to maintain their function if some of its damaged parts do not work. We need to find those intermediates to preserve the sight of patients. I always wake up in the morning with this vision: every child should have the right to sight. Thank you.